Okay, so welcome to this video. This is the first video in what I'm hoping is going to become a short series on the problem of obesity. Now, this video is intended for fellow medical students, but I'm hoping that it will be understandable to anybody. Because this is an issue that affects a lot of people in the world, and a lot of people would like to understand better. And in this series, my main point is going to be hopefully to try and persuade you that dieting probably doesn't actually work. I mean, if you didn't need any more evidence in a way, because look at the millions and millions of women who dieting does not work for, and they continue on thinking that the reason that it doesn't work properly is because they're not doing it properly. They just need to have a bit more willpower, and it's something that is drilled into us that dieting does work. If it isn't working, then you're not doing it right. But in this video, I'm hoping to show you that it's not in my opinion, it's not the right view. There is a bigger problem than just you eat too much. Fundamentally, you are overweight because you do eat too much. But, you are eating more, you are consuming more calories than you are burning is the fundamental problem. But the fact that you can voluntarily change that, I think we're hoping to show you in this video, is not the case, because you can, of course, you can withhold, you can fast, you can stop eating, and you will lose weight. But if you then stop dieting, you won't just, and you're at your lower weight, you won't just eat the right amount to stay at that weight. Your body will make you eat this enough until you get back to your original weight, and I'm going to show you the physiological mechanism controlling that, because there is a homeostatic mechanism which controls body weight, and basically it's as though your body has a fixed weight that it wants you to be, and it will make you eat the right amount such that your body weight becomes that fixed body weight. And the pathway, one of the central pathways to this, and the pathway which we're going to look at in this video, is the leptin pathway. Leptin pathway. Okay, and in the series we're going to do the leptin pathway, we're then going to do cholecystokinin and ghrelin, and we will talk about, along the way, how potentially these mechanisms go wrong in the pathogenesis of obesity. And it should be noted that except in very rare circumstances such as Prader-Willi syndrome, we don't actually know what the cause of obesity is in the general population. We do have noted that the leptin resistance is seen in a lot of people who are obese, and it does look as though that's a promising mechanism by which obesity could be generated. And then we'll finish the obesity series by looking at two drugs, sibutramine and orlistat, which are currently prescribed to help treat obesity, and we will discuss their mechanism of action. Okay, so the leptin pathway then. So to start with, we need to discuss what fat actually is. We'll talk about subcutaneous fat. So the fat on your belly, if you have a round belly, you'll have potentially layers of fat over your abdomen, and I'm going to describe the structure of that. So basically what you have, that's known as subcutaneous fat. So if you have the skin has many layers. So you have the outer layer of the skin is called the epidermis, and it sort of folds in like this. So this whole layer here is called the epidermis, and epi is either Greek or Latin. It means around. I think it's probably Greek. And dermis is just around the dermis, because the dermis is the layer underneath that. So this layer is surrounding the dermis, so it's around the dermis. So this is the outer world, so this is the environment up here. Environment. And there are multiple layers of the epidermis, so there's 5 layers. Sometimes people only say there's 4 because fifth one isn't distinguishable, but in some portions of the skin there are at most 5 layers. So here's the bottom layer, which consists of the stem cells of the epidermis, which are the only cells that are actually dividing in the epidermis, and they just divide and create new cells. So this layer is called stratum. Stratum just means layer. Stratum germinavitum, / it's also sometimes called stratum basal. Stratum germinavitum, both names make sense. Basal just means, it's at the base, and germinavitum means that it's germinating in some way, it's dividing. Then the next layer is called stratum spinosum, and it's because if you look down the microscope, all the cells start to become really spiny. They look as though they've got little spikes all over them, like little hedgehogs. So that's why that's called that. And remember that as the cells gradually get closer to the environment, they gradually die and become keratinized. So the outermost surface of the epidermis is dead cells, the layer that's actually in contact with the outside world. So as you go up the layers, the cells are gradually becoming keratinized and dying. So this is stratum germinavitum, not stratum germinavitum, stratum spinosum. Another layer above that is it's granulosum, stratum granulosum, and the cells become really dark in here. Stratum granulosum. The next layer is stratum pellucidum, or just stratum lucidum, and the cells become really sort of like they start really to die in this layer. They start to become almost transparent. Their boundaries break down, so they just look like one continuous layer now. So stratum pellucidum. So almost like a syncytium, stratum pellucidum. So you might see the comparison with necrosis here, but of course it's not but the cells are dying in effect. And this outer layer is the last layer is called stratum corneum, and here the cells are definitely dead, and there's loads of keratin strands everywhere. So that's what that layer is. So those are the 5 layers of the epidermis, and they are histologically distinguishable. The layer that's not always in all layers, that's not always visible, is the stratum pellucidum, but you always have the other 4 layers distinguishable. Okay, so underneath that you have the dermis, so this bit is called the dermis, and that's where all your sweat glands are. What else is down there? All your nerve endings, which sense things. So when I press on my finger here, there are nerve endings that are detecting that pressure, and they're all in this layer. You also have what else? Other things. Hair follicles extend into this layer, etc. So you'll have blood vessels in this layer, of course, as well, and lymphatic vessels and things like that. So this layer is really just the cells, and this layer has more sort of the other functions of the skin. So this is like the boundary, and this is the other functions of the skin. And then underneath that, you have a layer called the subdermis, or the hypodermis. And this is where all the fat is. There are loads and loads of fat cells in here. Now, a fat cell is just a cell like any other, which is absolutely stuffed packed with this huge globule of fat. So if this is the cell membrane, they are absolutely stuffed with this fat globule, and the nuclei are always pressed right up into one corner, if you like. Well, it's a circle, so it doesn't have a corner, but one aspect. And they often look down the microscope like rings with a jewel in. So that's one of the ways that you often identify them, because when you look down the microscope, the way that they fix fat cells, the way that they fix all histological tissue, means that the fat globule is actually washed away in the process. So you just end up with these huge white dots in the middle of the cells, which are the chromatin. When you stain it, it goes a beautiful pink colour usually. If you stain it with like eosin, they often stain it with. and the chromatin goes a beautiful pink colour. So you've just got this pink rim, and then suddenly a great white dot, and then this black sort of purple, often, if it's stained with hematoxylin, which it often is, this purpley, dark purple dot in one corner that looks like the jewel of the ring. So that's a way of identifying them down the microscope. And so you have this layer of fat which will differ in its size depending on how many fat cells you have. So if you have more fat, you will have a thicker hypodermis. And then underneath, you will obviously have connective tissue. So you'll have fascia, as it's called, which is just collagen. And you'll have connecting, because this is all just goo, really. So you have to have big trabeculae of connective tissue which attach the skin over here to the connective tissue below. And then underneath that, you'll start to have the muscles. So we're doing the abdomen, so we might as well discuss some of the muscles of the abdomen, the abdominal wall. The outermost layer of muscle of the abdominal wall, so what will be here, if you like, right under the fat, is called the, so if we view this as a cross-section, so if you've got your man here, and we take a cross plane through there, then that's what we're looking at present. So we've done the skin here, and now we're going further in. So the outer muscle is called the external oblique muscle, and it sort of goes like that. So if this is your going around like here, and if we go all the way back, well, the muscle doesn't go all the way back, so that's a bit misleading. But down back here, you'll have your vertebral column, like this. And this is a terrible drawing of the vertebral column. But you'll have a vertebral column, something like that, and a spinal cord, hopefully. And here is your muscle. So this is a cross-section, as I've shown here. So you have this external oblique muscle, which comes round like this, and then it stops. It turns into something called an aponeurosis, which is just basically a huge strand of connective tissue, so collagen all over the place. And then it continues on the other side, and you've got muscle. So you've got this layer of muscle, and the two halves of it are connected by just connective tissue. So imagine this is pink, and this is sort of like a grey colour of connective tissue. And that's pretty much the outer muscle layer. Then, sitting behind this muscle, you have another one pretty much doing exactly the same thing, which is called, sensibly, the internal oblique muscle. And this one gives off an aponeurosis as well, but the aponeurosis actually splits into two, like this, and I really wish I hadn't written muscles there. So it splits into two, so ignore the muscles. So this is the aponeurosis, splitting into two. Oh, and in fact, I've missed a bit out. It joins in the centre. So it rejoins up in the centre like this. So it creates these two compartments here, like that. So you've got these two compartments here. And if you think about it, which muscle is famous, a famous muscle that sits right at the centre of your abdomen in this central position, and you have two on either side? This is the rectus abdominal muscle, more commonly just called abs. Rectus abdominis muscle. And that's the one that people like to show off if they have a visible one. Rectus abdominis. And there are, as you go down, so if we draw it from the front, as you go down, so you have these two muscles in these compartments that are made by this aponeurosis of the internal oblique muscle. As you go down, at points, this doesn't split into two. Instead, it remains confluent. So let me draw what I mean by that. At some points, basically, this aponeurosis of internal oblique just remains as one bit, and that means that this muscle gets crushed in between them and creates what are known as tendinous intersections, and that's what creates the sort of segmented appearance of the abdominal muscles, what creates the six-pack or eight-pack. I think there are 8 components of it. So there's another one up here, kind of thing. And then they attach onto the ribs up here. It attaches onto the bottom 4 well, not the bottom 4 ribs, the bottom 4 ribs that attach onto the sternum. So not rib 11 and rib 12, we're not counting them. I mean rib 10, rib 9, rib 8, rib 6, which all sort of coalesce and join onto the sternum. So that's where rectus abdominis attaches. And behind that there's another muscle called the transversus abdominis, which again has an aponeurosis here, and it just sort of sits behind like that. And that's the abdominal wall then. So basically, back to what our original pathway was, the leptin pathway. And I want to keep this drawing, but I need more paper. We'll do it down here. Okay, so the leptin pathway then. This is a pathway for trying, it's a homeostatic pathway to try and make sure that you maintain a constant weight. Basically, what happens is that all of your fat cells create a certain hormone, if you like, a protein called leptin. And leptin goes up to the brain and basically tells the body how much fat you have got, because many fat cells you have determines how much leptin you make. So the more leptin you have, the more fat cells you must have. So it's the way that the brain knows how much fat the body has. And basically, the idea is, what we believe it does, and I'm going to show you the pathway by which we believe it does this, and this is still a bit of a contentious pathway. Leptin isn't fully understood. There is a lot of research going on to this. If we can find a decent explanation, if we actually find, what the cause of obesity, and we find a drug treatment for that, it will make billions. So there is a lot of research going into this. And of course it will help a lot of people as well, rather than the money. But this is not, it's not as solid as the anatomy is. The anatomy isn't changing. This pathway might be totally different in 50 years. So the fat cells here are making this product they're called leptin, and basically it goes up to the brain, tells the brain how much fat you have. It goes up to a specific bit of the brain, which I'm going to show. I'm going to show all this. We're going to go over all of this. This is just like a summary to tell you where we're going. And basically, your brain adapts how hungry it makes you feel, how much you want to eat, to how much fat you've got. So it takes in, if you've got more fat than your brain wants, it will make you less hungry. And this is kind of why, this might be why, when you binge over the Christmas break, so over Christmas holiday, people eat far more than they would usually eat, and their body weight goes up, they get more fat, so they make more leptin. And then when New Year comes, everyone diets, and everyone finds it easy to diet. Why do they find it easier to diet? Because after Christmas they have more fat cells, they are making more leptin, so their brain doesn't want them to eat that much. So they find it much easier to diet because their brain is on their side. Their brain wants, this homeostatic system is on their side. It's saying, don't eat more, eat less. So that's why people find it very easy to diet for the first few weeks of New Year, and then they break it because suddenly they go back to their original weight, and their body says, right, now you can eat normally again, and it's no longer working in your favour to diet anymore. So this system could explain that phenomenon that we all know, that when you've binged, you find it easy to diet because you kind of find food gross for a while. You don't want to eat cake, and that feeling of not wanting to eat cake, that feeling of not being hungry even though you know you haven't eaten for 6 hours, but you just don't feel hungry, that's because potentially because of this leptin pathway. Okay, so I'm going to show you the leptin pathway in more detail then now. So these fat cells have a nucleus that I have shown up here. This little black dot is the nucleus, and in the nucleus there is DNA. So I'll draw a little thing of DNA here. This is DNA. And somewhere on your DNA, you will have a section of this DNA called the Ob gene. Ob for obesity, Ob gene. because it was noted that if you take a mouse and you deliberately destroy this mouse's, obviously when the mouse was a single cell, when it had just been created through fertilization, what some scientists went in and did was they, well actually I don't know if they did this, it might have just been that it emerged. I think actually it did emerge. What they did is they inbred the mice, and eventually they found a mouse that, I think what they did is they inbred the mice, they found a mouse that got incredibly fat, and what they did is they looked for what genetic abnormalities that it had. And they found that there was mutations in this gene, and I believe that they must have by now done knockout experiments where they have actually taken a mouse zygote and knocked out both OB genes, because of course we are all diploid, we have two copies, we have effectively two identical pieces of DNA coding for this gene. Well, they might be slightly different, which is what gives you, is what you might have different alleles effectively. But if you knock both of those genes out, you get a mouse that is enormous, which is why it's called the obesity gene, because if you have a mutation in it, you become obese. Well, mice do anyway. And this gene, what this gene does is it makes leptin. So it produces, well, it firstly produces mRNA, so we'll draw mRNA, we'll just draw it as a line. mRNA will then go into and this is getting really too crowded, I want a new piece of paper. mRNA does not go into the abdominal cavity, it goes into the cytoplasm. So if we have this fat cell, and we have our huge globule of fat, and we have our nucleus, and in the nucleus we have our OB gene, and the OB gene produces the mRNA. So this is mRNA, this line here, messenger RNA. And the messenger RNA goes through the ribosome, which we'll draw like this. So the mRNA goes through here, and it spits out a polypeptide that comes out here, which folds into leptin, which I'm going to draw like this, because that is kind of what it looks like, well, it's a representation of what it might look like. So this is leptin, this sort of, it looks kind of like the Twin Towers, but this is meant to be leptin. Leptin, it's got that little joining there, leptin, and basically the leptin then leaves the cell, which I've conveniently drawn, and it is going to go into the bloodstream. So now time for a bit more anatomy, but before we do that, I'm just going to talk about some things that control this besides just the amount of fat. So one of the main things that controls how much leptin you produce is how many fat cells you have. If you have more fat cells, they're all producing leptin, you'll get more leptin. But there are other things that control how much leptin an individual cell produces. So, for instance, cortisol. Cortisol is a hormone. It's infamous as the stress hormone. When you are stressed, you produce more cortisol. But cortisol is a hormone that is produced if you become extremely hypoglycemic. Well, not extremely, but if your blood glucose goes below 4 millimolars, which it should not, unless you have diabetics, actually quite often diabetics on treatment with insulin often suffer this, because basically they inject into their muscle too much insulin and they don't eat enough. And then basically what happens is that the insulin goes too high and it causes blood glucose to go too low. So they don't manage to balance their food intake to how much insulin they have administered. Of course, they have to administer their insulin. If they're a type 1 diabetic, they must inject their insulin. There's no question of that, otherwise they can go into diabetic ketoacidosis, which is deadly as well. But hypoglycemia can be deadly as well. Diabetes, type 1 diabetes, is really, really an awful disease. It's diabetes is one of those diseases that people sort of don't realise how bad it is, because quite a lot of people have it. It doesn't command the sort of fear that cancer commands, but type 1 diabetes, it happens to children and it is awful. It can be really fatal if it's not managed properly, and it would be really fatal if it wasn't managed properly, but it can be managed very, very well, and you can do very, very well if you manage diabetes properly. But if they do become hypoglycemic, cortisol can start being released in huge amounts. So it signifies that blood glucose is too low. So it's clearly going to have some sort of role on the release of leptin by these fat cells. Cortisol basically is a glucocorticoid, and its structure, I'm going to draw its structure out now because it's got a steroid structure, and it kind of looks like this. So all steroids have 4 rings. So this is the first ring here, this is the second ring, this is the third ring, and this is the fourth ring, which is a pentagon. So you have 3 6-membered rings, and 1 5-membered ring. And off this position 3, which is very important, this is carbon free or position free, you have a carbonyl group. Off this group up here you have a methyl group. Off here you have a methyl group. Off here you have a hydroxyl group. Down here you have a double bond. And what else? Oh, you have a hydroxyl off up here. You have a carbon off there, carbonyl, and what then? Oh dear, what do you have off this? Is it just a methyl like that? That doesn't seem right. I'm just going to look up on Google the structure of cortisol because I want to get this right. I think there's something off here. So cortisol structure. The images. Yes, I'm right. There is something off there. There is a hydroxyl group coming off there as well. So an OH group comes off here. And cortisol is a glucocorticoid. It's the main glucocorticoid in the human. There are others in other animals. So there is corticosterone as well, but it's not that important in humans. Okay, so this glucocorticoid comes into the cytoplasm of the adipocyte. It binds to the glucocorticoid receptor alpha, so we'll draw that here. The glucocorticoid receptor alpha usually has another little protein bound to it, which is called the heat shock protein 90. And basically, when cortisol binds, the heat shock protein 90 cleaves away, and then what happens? So it's now on its own with cortisol attached. And then when you've got 2 of these, because this is now cleaved away, they dimerize. So you get a dimer of glucocorticoid receptor alpha. And this dimer then goes into the nucleus and acts as a transcription factor. And basically, the action of cortisol bound to this glucocorticoid receptor alpha is that when this glucocorticoid receptor alpha dimer goes into the nucleus, it binds to the OB gene and it promotes the expression of the product of this gene, so you produce more leptin. So that is why people are quite worried that actually stress might increase your risk of obesity because you are increasing your leptin. Oh no, because you no, cortisol. If you increase cortisol is going to increase your level of leptin production. If you increase your level of leptin production, then your brain might get It's believed to be a risk factor potentially for leptin resistance, that stress, which increases your cortisol level, is increasing your level of leptin inappropriately, and it might mean that your brain then becomes adapted to that level of leptin. So when the cortisol level goes back down, then the brain thinks that original leptin level was normal, and then you're going to then end up eating too much to try and increase your leptin release by increasing your fat cells. Okay, so that's cortisol's effect on this. Estrogen has a very similar effect. Estrogen is a very similar molecule, so if we draw the structure of estrogen, here you have a benzene ring here. I know the structure of estrogen, I think. And you have, well, actually, this is an interesting point. There is not just one estrogen. Estrogen is not just one molecule. There are 3 estrogens, and we're going to draw the main one, which is estradiol. So there are 3 estrogens. The other 2 are estrone and estriol, which play an important role, I think, in pregnancy. Estriol, I think, in particular. And it basically has the same steroid structure with the 4 rings. This is a 5-membered ring. And off this one, instead of having this fancy group that you had up here, you just have a hydroxyl group. So you have these 2 hydroxyl groups, which is why it's called estradiol. So estradiol. You can't have the methyl group there because you've got this benzene structure, estradiol. So all that, the valencies of that carbon are full. And that's the structure of estradiol. So estradiol basically comes in and does the same thing. It binds to an estrogen receptor, which is cytoplasmic estrogen receptor. The estrogen receptor then translocates into the nucleus and acts as a transcription factor to increase the expression of this gene so that you produce more mRNA and then you produce more leptin up here. So you produce more leptin overall. Other hormones that affect this are insulin. So when you have high insulin levels, insulin can also increase the expression of leptin and therefore increase your leptin. Why would you want to do that? Well, insulin, if you've got very high insulin, that signifies that your blood glucose is very high. Usually, if you're not diabetic, it signifies that your blood glucose is very high. So it signifies metabolic well-being. So if insulin increases leptin release by the adipocytes, then leptin is going to go up and tell the brain, stop eating, which is appropriate because you're in a state of metabolic well-being, so you don't need any more food. Okay, so that's insulin, and also there's adrenaline here, noradrenaline, that plays a role. So basically what it's found, adrenaline and noradrenaline here, so it's found that if you stimulate beta adrenergic receptors on these adipocytes, so if you have a beta adrenergic receptor and you stimulate that, it inhibits the release of leptin. Why might you want that? Well, adrenaline signifies that you're about to go into fight or flight. So it signifies that you're about to use a lot of energy. So you want, after you're obviously out of the situation where you need to fight or flight, you want hunger. These are obviously all speculations. We don't know this. But we speculate that once you have completed your fight or flight, you are going to need to consume some more food to make up for the energy that you have lost in fighting or flighting. And basically, this is going to produce that result, that if noradrenaline lowers your leptin level, you're going to get more hungry. Okay, so those are the influences on this. So now let's do the pathway by which leptin gets to the brain. And for this, we probably need our diagram here back again. So we were looking at the abdomen. So leptin we'll draw here. This is the leptin, and it is coming out of the adipocytes. So pathway that this might get to the brain is going to be through it's going to have to go through a vein. So there is a nice vein here in this running behind rectus abdominis. So if you go into your abdomen, you go through the skin. So if you go through the skin up here, you go through the epidermis, you go through the dermis, you go through the layer of fat, you get to the aponeurosis of external oblique. You go through that, you go through the anterior rectus sheath, that's what this bit's called. This bit in front of the rectus muscle is called the anterior rectus sheath. Anterior rectus sheath. You then have the rectus abdominis muscle here. Behind that you have, guess what, the posterior rectus sheath. And in front of the posterior rectus sheath, but behind the rectus abdominis muscle, you have a vein here, called the superior epigastric vein. Superior epigastric vein. Epigastric vein. And we're talking about up here sort of level. There's another one down here called the inferior epigastric vein. But it's up here that we're going to look at. So up here, we've drawn it nicely there. This is the superior epigastric vein. And basically, it goes up in behind the rectus abdominis muscle, but in front of the posterior rectus sheath, and it then gets to the ribs up here. So this might be the boundary of the ribs and the sternum there. And basically what's going to happen is it's going to go behind the ribs, and now I think I probably need a new picture. So if we have the sternum here, so the sternum here, and you have the zippy sternum at the bottom there, then you have sort of like this, you have a bit coming off like that, it splits into the bottom 4 ribs, kind of like this, maybe not quite as extreme as I'm drawing it, but it gives off the bottom, no, well not the bottom 4 ribs, but the bottom 4 ribs that are attached to the sternum. There are another 2 ribs called floating ribs, which don't actually attach to the sternum, so that's 11, 12, this is 10, 9, 8, and 7. And then 6 comes off up here, it gives off like a branch, I think, no, not like that. I think it's like that, don't quote me on that, but I think it goes like that. And that's going to be 6, and it will go on upwards. So off this, you have the rectus abdominis, more like that, coming off down there, and the other one on the other side. And then we have, we'll let this be the superior epigastric vein, so I was just being fortuitous, and it goes behind these ribs, and it goes up, and there's still ribs out here that it's behind, etc. And it goes up until you get to like the manubrial sternum up here, and how many ribs have I drawn? 5, 4, 3, we'll have 1 up here, and 2, and it goes up like that. You've also got the clavicle here, and it goes up basically. And where does it join onto? Well, if we draw the circulation for what feels like the umpteenth time, you have the what do we have? Okay, right, so you have the circulation would be up here. I should have drawn this further down so that I could draw the circulation on it. But up here you'll have the formation of the superior vena cava, and this vein basically joins on somewhere up there. So if we draw the internal jugular vein joining together with the subclavian vein, in this case the right subclavian vein, and this is where the superior epigastric vein joins on. So just before these 2 brachiocephalic, or sometimes called innominate, veins join to form the superior vena cava. And here is the same thing on the other side. So this is the internal jugular. These are the internal jugular veins, right and left. This is the right subclavian vein, the left subclavian vein, the right superior epigastric vein, the left superior epigastric vein, the brachiocephalic veins, or the innominate veins as they're sometimes called. I think surgeons like to call them the innominate veins. And this is the superior vena cava. Coming down to join onto the right atrium, which then goes into the right ventricle, which pumps blood out into the pulmonary trunk, and then you have off here, maybe we'll draw, so we'll have off there like that. Yes, this is going reasonably well at this drawing. There shouldn't really have that there, and then you'll have the aorta coming off up here. So that's the left atrium. This is the left ventricle, the right ventricle. So off here comes the pulmonary trunk, which gives off the, start again. Blood comes in, so leptin is coming up through the superior epigastric vein. It's going into the brachiocephalic veins, into the superior vena cava, into the right atrium, into the right ventricle, up into the pulmonary trunk, and then into these 2 pulmonary arteries I've drawn here. This pulmonary artery, this is the right pulmonary artery, goes behind the ascending aorta, which I have drawn rather awfully there, and then it goes into the lung. So leptin is going to go through the lung, never mind. It's going to go into the pulmonary capillaries, it's going to go straight through them, and it's going to come back into the 4 pulmonary veins here. So these are the ones draining the left lung, and these are the ones draining the right lung. So leptin comes back in, it goes into the left ventricle, and then it comes out of the aorta, and then it goes upwards. So I'm going to redraw. Can we continue on drawing it here? Okay, we will do. Actually no, we won't. We'll redraw the aorta. Okay, do it on the back of here. So, right, so if we have the ascending aorta like this, and it goes around into the aortic arch, off the first branch to come off the aortic arch is the brachiocephalic artery. Then we have the left common carotid, the left subclavian, which isn't of interest to us, and these are the 2 main ones, and this branches off to give the right common carotid, and then we've got the right subclavian here. So these are the 2 arteries that are important to us in this story. So the common carotids, carotid arteries. Okay, so leptin is coming out of the aorta and it's going up these 2 arteries. And these 2 arteries ascend in the neck. They are within the carotid sheath. They ascend medially in the carotid sheath. They are medial to the vagus nerve, medial to ansa cervicalis, medial to the internal jugular vein. So should we do a bit of neck anatomy? So if we've got our neck here, these 2 arteries ascend kind of here. And if we take a cross section through, so this is if you've got your head going up here, this is looking at you as I would do. Then we're going to take a cross section through, and it kind of looks like, well, it obviously looks like a circle. And at the front you have, well, actually we'll start here kind of thing. You have the esophagus here, which is the pipe down which your food goes. It's got sort of like a ring of connective tissue around it. In front of that you have the trachea here, which has a layer of muscle at the back called the trachealis muscle, and it's sort of a cartilage structure like that. And then around that you have the thyroid gland, kind of like that. So these are the 2 sides of the thyroid gland, and they're connected in the middle. These should probably be a bit bigger, more like that. And then down here kind of thing, well, you'll have your vertebral body and the rest of the vertebra down there. And down here you have the carotid sheath. And in this you have, here is the common carotid artery coming up. You have a huge great vein, which is the internal jugular vein. And then sitting here you have the vagus nerve. And sitting here at the front you have ansa cervicalis, which is basically it's a nerve that controls most of the muscles of the neck. So that's the anatomy of the neck. So it's going to come up these arteries up into the head, and it's going to get the common carotid artery is going to branch. It's going to give off the external carotid, and it's going to give off the internal carotid. The internal carotid then goes up further. It goes through the carotid foramen, through the carotid canal, and I should try and draw this at least. So if we have the middle cranial fossa there, then you kind of have the carotid canal, which isn't visible, but it's going to come up through here. It's going to come out of a hole into the middle cranial fossa, and then it's going to go this sort of way. It's going to go over the foramen lacerum. Remember, basically nothing goes through the foramen lacerum, so it just sort of turns over it and then goes upwards. It goes through the cavernous sinus, so we'll have to put the cavernous sinus here. It goes through that, and then it goes into the circle of Willis, which we're now going to discuss. Can I do it on here? I think I'd prefer a new sheet of paper. So we'll now discuss the circle of Willis. Okay, so we're now going to look at the brain from its inferior surface, or its ventral surface, because the brain's all weird with regards to which side is ventral and which side is medial. So if we have the brain drawn here, this surface down here is said to be the ventral surface. So we're going to look up, we're going to imagine we're down here and we're looking up at the base of the brain. And specifically we're looking right at the center down here, which is where our artery, our internal carotid artery, is going up to. So basically we're going to look at the base of the hypothalamus. So here is our two arteries, our two internal carotid arteries, which sort of turn this way, and they eventually become the middle cerebral artery. So it's sort of coming up, and then it's turning. So this coming into the page from where you are positioned, we're now looking up this way. So here are the internal carotid arteries which have just come up, so they'd continue on like this, downwards. So these are the internal carotid arteries. Can I label them? I will do. Internal carotid arteries. Internal carotid arteries. Yes. And then over here, down here we have the pons. And on the surface of the pons, you have the basilar artery. So this is the basilar artery. Basilar artery. And the basilar artery at the level of the midbrain, well, before the midbrain actually, it splits into the two posterior cerebral arteries. So these are the two posterior cerebral arteries here. And these give off branches called the posterior communicating arteries, which go out here. So those gaps there shouldn't be there, those lines there shouldn't be there. There should be a gap here and a gap here, and a gap here and a gap here. So there's a branch from these two. These are the posterior cerebral arteries. Posterior cerebral arteries. And this is the posterior communicating artery. Communicating artery. Communicating artery. Communicating artery. And then you have kind of in the position here, you have the optic chiasm. So here's the optic chiasm giving off the optic nerves. So these are the nerves which go to your eyes and carry the visual information back to the brain. And they swap over. So half of the nerves, half of the axons running in this optic nerve will go into this optic tract. And these ones will cross over into this side, and these ones will cross over into this side. And the half here will stay on this side, and the half here will stay on that side. So that's the optic chiasm. And this then gives off the anterior cerebral artery, which goes behind, it goes up, it goes dorsal to the optic nerve, and then turns on and gives blood, rather, doesn't innervate, it gives blood to the anterior cerebral, the anterior portion of the brain. And there is a small communication between them, so there's a little artery there, so if we fill them in with blood so that you can actually see them, that communication is called the anterior communicating artery. So now you can see the full circle here. Anterior communicating artery. Communicating artery. Communicating artery. Okay, so what do we need to do now? Okay, so this becomes, by the way, this now is the middle cerebral artery over here. Middle cerebral arteries. Cerebral artery. Okay, so at this space here is the hypothalamus. Well, there'll be a midbrain here, but you can't really see the midbrain. It's sort of more down there. And here what you have is two sort of balls that hang down, and these are called the mammillary bodies. And I don't know why they're called that. It's probably because they look like the mammary glands, but they have nothing to do with lactation. They're actually very important in memory. And if you get lesions to them, you get a form of amnesia similar to medial temporal lobe amnesia, called diencephalic amnesia. And you have symptoms similar to HM, where you have an intact short-term memory but no long-term memory, and you have an intact procedural memory, so you have no long-term. So yeah, sorry, I've made a mistake there. You do have some long-term memory. You have procedural long-term memory. So if you didn't know how to swim or something, I could teach you how to swim even if you had this sort of amnesia. But with regards to trying to teach you facts or trying to get you to remember what you ate yesterday, you can't do it. You have no episodic or semantic memory. You have lost declarative long-term memory, but you have intact non-declarative long-term memory. So you have intact procedural memory, and you still show Pavlovian conditioning with regards to fear responses, etc. Okay, so these are the mammillary bodies, and as I say, they're important in memory. And there is a sort of bulge here called the tuber cinereum, and off the top of this you have the pituitary stalk. So in front of here you'd have the pituitary gland, sort of like that. Well, not like that, like that. But so you have the anterior pituitary and the posterior pituitary, but they'd be hanging down, and I shouldn't have really drawn them. They're not important with regards to our discussion here. So off here comes a little branch. Well, there are two branches actually. There's one here, and there's one, so there's this one on the other side, and there's another one here. And these two branches are called, so they come off the posterior communicating artery here. This one is called the chiasmatic branch, going to the optic chiasm, and this one is called the hypothalamic branch, going to the hypothalamus. And this is the portion of the brain where the leptin is going to. So leptin comes up the internal jugular, not the internal jugular vein, the internal carotid artery, into the posterior communicating artery, into the hypothalamic branch, and right on target it goes to this bit kind of here, which is a specific nucleus of the hypothalamus called the arcuate nucleus. So, arcuate nucleus of the hypothalamus. Arcuate nucleus. And this is basically the core of leptin now. We've done all of this anatomy to show you where leptin comes from, where it goes to, and how it gets there. But now, what's it going to do here? How is it going to produce that feeling of satiety, that feeling of being full? Well, our honest answer is we don't really know. We know that it acts on this arcuate nucleus, and I'm going to give you a bit more information about that. But how a molecule acting on a few neurons actually produces a conscious experience of being full, we don't have a clue, because we don't know how a goo of cytoplasm and action potentials and all of this creates a conscious being. What we do know is that this does seem to be a mechanism by which you can produce satiety. So the mechanism is going to be quite black boxy after a while, but I can't really do much more beyond that, I'm afraid. Okay, so in the arcuate nucleus then, there are two types of neuron. So, neuron type 1 and neuron type 2. And one type of neuron is called orexigenic neuron. Orexigenic neurons. And the other type of neuron is called anorexigenic neurons. Anorexigenic neurons. And basically, your level of hunger is believed to be controlled by the level of activity in these two neurons. If these ones are more active, you become hungry. If these ones are more active, so it's like a seesaw. The more one is active, it's a balancing act. If this one dominates, if this one is overactive and this one is underactive, you become hungry. If this one is overactive and this one is underactive, you become full. So that's why anorexia, the feeling of not wanting to eat, and orexigenic is kind of the opposite of that. So basically that's it. Leptin comes up to the hypothalamus, activates these neurons, and seems to inactivate these neurons and tips the balance towards the anorexigenic neurons being dominant. And that somehow, through a black box, makes you feel full, we believe. And what we're going to see is that other systems, such as ghrelin and cholecystokinin, both work also by doing this exact same thing, or by interfering with this. I'm going to give you slightly more information about these two types of neuron now, and I'm then going to tell you about the drug sibutramine, because it acts straight on this nucleus, we believe. To try and make this one more active and this one less active. So what was I going to tell you? Okay, I was going to tell you about some properties of these neurons. So when you look at these two different types of neuron, how do you know if you've got a lump of arcuate nucleus? How do you know which neurons are orexigenic and which ones are anorexigenic? Well, they have different chemical properties. These ones contain certain neurotransmitters, and these ones contain a different type of characteristic neurotransmitter. So the characteristic neurotransmitter of anorexigenic neurons is what? What can I remember? It's POMC, which stands for this is a mouthful, pro-opiomelanocortin. Melanocortin. So there is a lot of this stuff in these anorexigenic neurons. And what is this stuff? Well, actually, the reason it's got this pre-pro there is that it's the precursor to lots of other stuff. So the reason it's got opio in there is that it's a precursor to beta endorphin, which is an endogenous. The reason it's called endorphin is it stands for endogenous morphine, and endogenous morphine, and morphine is an opioid. And it's actually more than an opioid, it's an opiate, so it's actually got the chemical structure that characterizes an opiate. So just a little biochemistry check here. An opiate is a chemical structure. To be an opiate, you have to have a certain chemical structure. So not cocaine, sorry. Cocaine is not an opiate. Morphine and heroin and codeine are opiates. An opioid is a more general thing. An opioid is anything that acts on opioid receptors. And opioid receptors are named after the opiates, because basically what we did is we went in and we tried to find what do heroin and morphine actually act on. And we found these receptors and we called them opioid receptors after opiates. And now the general term opioid is any substance that acts on these opioid receptors. So opiates are all opioids, but not all opioids are opiates. Okay, so pre-pro-opiomelanocortin is a polypeptide that you make, and then basically you cleave it, you break it up, you chop it up, and then from all the little fragments you get, you make other stuff. So it's a huge molecule from which you can make loads of different things. So for instance, this is its structure. It has a signaling chain here. So this is a signaling bit. So that's just important for making sure the molecule goes to the right place. Signaling molecule. Then you have a bit of junk. Then you have something called gamma melanocyte stimulating hormone, gamma MSH. You then have more junk. You have two bits that together, this whole fragment here, makes ACTH. And then after that you have beta endorphin over here, endorphin, and you have gamma lipotropin, LPH, and this overall bit here is called beta lipotropin, and this bit here is called CLIP, and this bit here is called alpha melanocyte stimulating hormone. So basically, this is the case. You can chop it here, and you can make ACTH, but you don't have to stop there. You can chop the ACTH up and make alpha MSH and CLIP. ACTH is obviously very, very important. It's adrenocorticotropic hormone. It's released by the anterior pituitary, and it tells the adrenal glands to make more cortisol. Alpha melanocyte stimulating hormone and gamma melanocyte stimulating hormone, I don't know what they do. I presume they stimulate melanocytes, which are the cells in the skin which make melanin. It's what gives you a tan. So they'll have some sort of role like that. And I don't know what lipotrophic hormone do either, but beta endorphin is very important in natural reward processes. So this seems to be kind of linked with eating, because we do find eating rewarding, and this potentially is why. Another neurotransmitter which is characteristic of the anorexigenic neurons is what's the other one? Oh, it's CART, standing for cocaine amphetamine regulated transcript. And this has quite an interesting structure. It kind of looks like this. It has like a hoop, and then the chain folds back through the hoop. So that's what that is. Cocaine, I'll write its name. Cocaine. I don't know what it does. It's just a molecule to me with a funny shape. Cocaine amphetamine regulated transcript. Regulated transcript. And then we'll do the anorexigenic neurons. So what's in the anorexigenic neurons? Well, you have neuropeptide Y, which is one that continuously comes up in medical school, but you never really find out what it actually does. It's just all over the place. Neuropeptide Y, and that one's structure is kind of like a spiral. So it's an alpha helix of a protein, and then it becomes like just a fat sheet. So that's its sort of its ribbon structure. And then what's the other one that's characteristic? Agouti-related peptide. Again, I don't know what it does. Agouti-related peptide. But I can give you a picture for it. It has a sort of structure. It has a flat bit in the centre, and then it has two sort of rings on either side, which is quite nice. It's kind of like a bone, but these are hollow, so you've just got a ring, and then this sort of solid bit in the middle. And that's what you get if you Google agouti-related peptide. So that is how, if you really wanted to, you could determine which neurons are orexigenic neurons and which neurons are anorexigenic neurons. Okay, so I just want to briefly say how the leptin pathway, I already have sort of hinted to this, but how the leptin pathway might be involved in obesity. Basically, what we think, this is a theory of obesity, we think that people who are obese have leptin resistance. Their body thinks they need, their hypothalamus is set to receive it wants to receive too much leptin, and if it doesn't receive enough, it's desensitized effectively to leptin. Leptin should activate these and inactivate these to make you feel full and stop you eating. We think the hypothalamus becomes resistant to leptin. It becomes unsensitive to it. It stops listening to it. So basically, you remain hungry, and then you put on weight, and your hypothalamus only starts listening when you get massively obese and you have a huge amount of fat, you have a huge amount of leptin, and then finally that massive amount of leptin is enough to stop the hypothalamus making you eat. So that is called leptin resistance, and we do see leptin resistance quite commonly. It's not something that you can test for. Well, I don't know if you can test for it, but it's not something I've ever seen a test being done for. I don't know even how you would test for it. But it is one of the characteristics that is put in metabolic syndrome, which is this syndrome that of basically when you, if someone is massively obese, they tend not just to have obesity, they have a whole sort of bunch of diseases that all sort of go together, and they've all been grouped together under this one term, metabolic syndrome. So obesity is one of the characteristics. CNS leptin resistance is another. You also see type 2 diabetes generally, inflammatory diseases, cardiovascular diseases, high blood pressure, and I think there are more, but that's metabolic syndrome, and CNS leptin resistance is counted as one of the syndromes of that, along with obviously insulin resistance under type 2 diabetes as that symptom. So have I said everything I want to say? No. Sibutramine now. The drug sibutramine. So the first of our two drugs. There are pretty much only two drugs that are used clinically to make you lose weight. There are incredibly powerful agents that will make you lose weight, such as dinitrophenol. The problem is they'll also turn you yellow and then kill you. Sibutramine is not a drug that will kill you and is actually licensed for clinical use. So sibutramine. So we'll start with its chemical structure because it's nice to have a picture for it rather than just a name. So you have a benzene ring, I think, and I should have checked this before I do it, but I think you have a benzene ring. I will check this in a moment. And then you have a chlorine atom off the side there. You have like a cyclobutane group, which is quite interesting. It's not something you see every day. And then you have a carbon coming up here. You have a nitrogen off here, and off this nitrogen you have two methyl groups, which I just draw like that. So it's a skeletal structure. And then you have an ethyl group coming off that, and then two methyl groups coming off that. And I will just once again check on Google that that structure is correct. Sibutramine. Images. Oh no, it's not showing me one. Yes, it is a benzene ring. Excellent. That's fine. Wait, where's the nitrogen? Oh yes, there it is. Yeah, that's fine. Okay, so how does this drug work then? Well, basically, it goes into the hypothalamus, and all I know is it blocks reabsorption of. I don't know, what am I doing? Reabsorption of two neurotransmitters, of specifically serotonin or 5-hydroxytryptamine and noradrenaline, which is quite interesting because that's usually what antidepressants do. There are many antidepressants that work by blocking, well, we think they work. We aren't really sure what the cause of depression is, so can't be sure that our drugs work in this way. But we think they work by blocking serotonin and noradrenaline. So, for instance, the tricyclic antidepressants, so amitriptyline and imipramine, etc., things like that, they work by blocking both. They generally block both noradrenaline and serotonin reuptake. I mean, there are other drugs such as the selective serotonin reuptake inhibitors, the SSRIs, such as fluoxetine, more infamously known by its trade name Prozac, which just blocks serotonin reuptake. But this drug, sibutramine, it works, we think, by blocking the reabsorption of serotonin and noradrenaline in the hypothalamus, and that somehow, the increased levels of these two neurotransmitters somehow increases the activity in the anorexigenic neurons and lowers the activity in the orexigenic neurons, makes you feel full, stops you eating as much, and therefore helps you to lose weight. So that is how we believe this drug works. It is mildly effective. You can Google before and after pictures. To be honest, if you take it and don't do anything else, you're probably not going to have that much success. But it does produce some results. I think it has been demonstrated in numerous clinical trials to be efficacious compared to a placebo. And since we've got nothing better, it is a good drug. So I think that pretty much concludes my leptin video. And what I said at the start is about how I don't believe dieting really that works. And the reason that I don't think it works is that if you have CNS leptin resistance, the problem is that your body has this set weight that it wants you to be, which is controlled by how much leptin the hypothalamus wants to receive in order to make you stop feeling hungry. And if you put yourself through, a terrible diet like the Atkins diet, which is one of the only diets that has been scientifically shown to actually work, and it's horrific. It makes a lot of people very, very ill going on that diet. I would not recommend it at all. But if you do go on a diet and you then come off it, the problem is that you're just going to go back to the same weight you were originally, because your body has this system, and basically it's going to mean that unless you're prepared to, for the rest of your life, suffer feeling hungry continuously, that basically you're just going to have to diet continuously. You can never eat until the point that you are actually full, because the system is trying to turn you back to your original weight. And that's why I don't believe dieting works. I may be completely wrong, but I think, I mean, you just have to look at how many people dieting does not work for to know that there's something wrong here, surely. So what do I believe is the cure to obesity? I believe exercise. Exercise. I think that is the cure to obesity. I don't know how, but I think somehow lack of exercise is what is contributing to leptin resistance. That's all speculation. I don't know that, but I believe that if you want to lose weight, this is the central message of this video. If you want to lose weight, take up exercise, and if you are not currently overweight, take up exercise anyway to prevent yourself becoming overweight. So thank you for that. I hope you have learned a lot, and I will continue this series. We will then look at cholecystokinin and ghrelin's effects on this same system, these orexigenic and anorexigenic neurons in the hypothalamus, and we will also discuss the other drug, orlistat.